Hello students, welcome to the biology class. So in the last class we learned about how nucleotides are finally formed. We learned that there are two types of major bonds involved in the nucleotide formation. One of the bond is the N-glycosidic bond and the second bond is the phosphoester bond. So two bonds are involved. I found students writing it wrongly. Nucleoside has only one special bond and that is the N-glycosidic bond, and the nucleotide has both N-glycosidic bond as well as the phosphoester bond, this bond. So what are nucleotides? We learned that they are phosphorylated nucleosides. So they are formed by attachment of the phosphoric acid group to the nucleoside. And what is nucleoside made up of? Pentose sugar and the nitrogen base. When they join together with a bond which is between the first carbon and the first nitrogen, or the first carbon and the 9th position nitrogen, these bonds are fixed in pyrimidine between first position carbon and the first position nitrogen. In the case of purine, it is between the first carbon position and the 9th nitrogen position. It is called, these bonds are called N-glycosidic bond. These bonds are called N-glycosidic bond, and then these bond formation cause the formation of nucleoside. And when nucleoside joins with the phosphoric acid, it turns into nucleotide. Then we learned the importance of the cyclic AMP. Cyclic AMP is the secondary messenger. Why secondary messenger plays an important role? Because there are some neurotransmitters. And there are some hormones which are not able to go across the plasma membrane and bring change inside the cell. We all know hormones play an important role. So if the hormone is not able to go across the plasma membrane, then how will it cause the changes? So the answer is secondary messenger, because hormones are called as primary messenger. So these messengers will be talking on the surface of the cell, and then there are receptors on the surface of the cell membrane. Then these receptors will start another chain of reaction inside the cell with the help of cyclic AMP. So cyclic AMP takes the message inside the cell and will bring changes, and finally the cell is going to show changes which are required according to the hormone which has brought the message. So this is how cyclic AMP plays an important role. We learned the differences also between DNA and RNA to some extent. Then we learned about the various functions of cyclic AMP. We learned about the nomenclature in the last class. So today we will start with the next topic, and that is the how higher nucleotides are formed and what exactly is the ATP, one of the most important molecule because it is the energy currency for us. So how higher nucleotides are formed. So in the last class we only learned about MPs. What are MPs? Adenosine monophosphate, or deoxyadenosine monophosphate, or guanosine monophosphate, deoxyguanosine monophosphate, cytidine monophosphate, deoxycytidine monophosphate, uridine monophosphate, and this will be called deoxythymidine monophosphate. So we only learn about the monophosphates in the last class, but if you remember ATP means adenosine triphosphate. So what exactly is possible is, instead of having just one phosphate, it is even possible to have two phosphates attaching with the nucleoside. Nucleoside means that pentose sugar, it can be ribose sugar or deoxyribose sugar, and the purine and pyrimidine attached, A, G, T, C or U. So it is possible that it could be one phosphate attached, or it can be two phosphates attached, or it can be three phosphates attached. So then it will be called as the higher degree of nucleotide formation. So let's understand how the other phosphates attach, and then what exactly is ATP. So ATP is called as the energy currency or the energy depot. So who discovered ATP? Remember this name, Carl Luhmann, a German scientist. In 1929, he discovered the presence of ATP, but the full function of ATP or the importance of ATP and that it is present in all the living contents, living organisms, whether prokaryote, eukaryote, everywhere ATP is present. So its importance was